Hello, everyone, and welcome to the 31st INFOGEST Viva Webinar. This webinar series is co-organized by the INFOGEST Research Network and by Teagasc and by our team of postdocs here in Teagasc. So today I will be here with my colleague, Shannon Gwala, and our speaker today is Wang. Welcome, Ricky. We are delighted to have you today. Thanks very much for joining the webinar and accepting to present your work. So Ricky completed his PhD at the University of Borås in Sweden. His PhD project aimed to investigate how fungi cultivation strategies affect their nutritional quality, particularly protein and also the mineral content. And he has also evaluated the digestibility of different edible filamentous fungi. And the title of his presentation is In Vitro Gastrointestinal Fate of Edible Filamentous Fungi, Protein and Mineral Digestibility for Food Applications. So, Ricky, the floor is yours.
Hello everyone, thank you for the introduction. Today I would like to speak to you about my PhD thesis on in vitro gastrointestinal fate of edible filamentous fungi with protein and mineral digestibility for food application. I would like to start with a quote from Nordic Food Innovation Summit that food is both the problem and the solution. About 1/3 of the total carbon emission, 70% of freshwater usage, and 90% of the global biodiversity loss can be attributed to the global food system. Most international reports, such as Nordic Nutrition Recommendation, Eat Lancet, and IPCC, have agreed that we eat too much red meat, and we need to reduce our meat intake and eat more plant-based protein. However, in this recommendation, we often do not include that some other novel protein sources. In this case, which is fungi, that is not animal or not a plant, but they are a separate kingdom of life. We know fungi as food, as mushroom. We have eaten mushroom for a very long time, and mushroom are the fruiting bodies of fungi. But what we cannot see normally are the mycelium, which are under the soil, and we don't normally eat them. There are some species of fungi that do not produce these fruiting bodies. We refer to them as filamentous fungi. And this filamentous fungi has been used in several traditional fermented food, such as tempe and oncom in Indonesia and koji from Japan. The idea is, can we grow this filamentous fungi in a bioreactor and we make a meat alternative out of the fungi? Filamentous fungi as meat alternative has several benefits. Firstly, it has the filamentous structure, meaning that this filamentous structure resembles much of the muscle fiber, which makes fungi has a good texture. Fungi are also a decomposer in nature, meaning that it can use a complex substrate, such as agriculture substrate. Fungi are also high in protein content, and they do not contain any antinutrients such as phytate. The commercialized fungi alternative are corn, which are already available in the market. And nowadays, there's a lot of startups also trying to commercialize this fungi-based meat alternative. Although a lot of work has been done on making fungi meat alternative, not much work has been done on the nutrition of the fungi. Particularly, what happens when we eat the fungi, how are they digested in our gastrointestinal tract? These are the questions that I would like to answer in my thesis. Food nutrition consists of protein, minerals, lipids, starch, vitamins, and dietary fiber. I focus on the protein and mineral aspect of the fungi because not all protein are equal. And the question is, can this fungi protein be digested by our gastrointestinal tract? For the minerals, we need to be aware of the antinutrients because like about 30% of women globally suffer from iron deficiency anemia, and this iron deficiency risk will only increase as we have the dietary protein shift to a more plant-based protein. So the aim of my study is to investigate the protein digestibility and the mineral accessibility of the edible filamentous fungi. In my thesis, I formulate several research questions. Firstly, how does the protein digestibility of fungi, which they have the cell wall structure, compare to that of animal-based protein? And also, how do the cultivation condition will affect the fungal cell wall and their protein digestibility? This part, however, I will not present in this presentation. And then how do the media mineral concentration affect the fungal mineral accessibility? And what happens if we grow the fungi in a media containing phytate, such as oat media? Will they affect the fungi mineral accessibility? And then if we grow the fungi in media with polyphenols, such as winery side stream, will they affect their protein and mineral digestibility? To do that, I use 5 different species of fungi, like Neurospora intermedia, Aspergillus oryzae, and Fusarium venenatum. Those are from the phylum of Ascomycota, and 2 species of Rhizopus, Rhizopus delimar and Rhizopus oligosporus, from the phylum of Mucormycota. The difference between those 2 phylum are their cell wall. For the Ascomycota, the cell wall are made of beta-glucan and chitin, while for the Mucormycota, the cell wall are made of chitosan and chitin. For growing the fungi, I grow them in the submerged fermentation using glucose media, oat flour, and winery side stream. And I cultivate them in different scale of bioreactor, from the shake flask to 4-liter bubble column reactor, and also a 26-liter bubble column reactor. To evaluate the digestibility, I use the INFOGEST method. And for the oral phase, with the salivary amylase, and gastric phase with the pepsin, and for the intestinal phase, and with the pancreatine. I modify the INFOGEST method using a less amount of enzyme for the oral phase and also for the intestinal phase. After the digestion, I measure the degree of hydrolysis, and then also for the amino acid accessibility and the mineral accessibility. First, I will discuss about the fungi protein digestibility. I study 5 species of fungi cultivated in the glucose media, and compare with the animal-based protein. And if we see that for animal-based protein, most of the protein content are higher than 50%. While for the fungi, the protein content are mostly lower than 50% at the range of 30 to about 45%. Although they have the lower protein content, if we see their essential amino acid, fungi have a very rich in essential amino acid, with some species are even higher compared to the animal-based protein. Fungi are also particularly rich in lysine at up to about 13% of the total amino acid. And then the question is, how is their protein digestibility? We hypothesize that because fungi has the cell wall, maybe it could have the lower protein digestibility compared to animal-based protein, which do not have a cell wall. However, if we see after the oral digestion and gastric digestion, the degree of hydrolysis of fungal protein are higher compared to animal-based protein. And so after the intestinal digestion. However, this high amount of degree of hydrolysis is mainly because of the high amount of primary amine that is already present in the sample prior to the digestion. To be able to say that the protein is digested by our gastrointestinal tract, we need to normalize this initial primary amine. And by doing so, we can see that the fungi protein digestibility comparable to the animal-based protein. While with Fusarium venenatum, slightly lower, but it is mostly because of the high amount of primary amine. Then I studied fungi mineral digestibility. I used 4 species of fungi cultivated in the glucose media. You can see that for the iron content of fungi, it ranged from 3 milligram to about 5.2 milligram of iron. While for the zinc, it could range from 8 milligram up to 20 milligram of iron. How about their accessibility? If we see for Neurospora intermedia, all of the iron, 4.8 milligram of the whole iron, are accessible. While that's also the case for Aspergillus oryzae, all of the iron are accessible. While for the Rhizopus dilemma, only 2.7 milligram of the iron are accessible compared to that of the total of 5 milligram. And so too for the Rhizopus oligosporus, which have the lower iron accessibility. For the zinc, Neurospora intermedia and have 52% of zinc accessibility, and Aspergillus has higher at 77% of accessibility. And for the Rhizopus, they have a lower amount of zinc accessibility. It seems like for the phylum of the Rhizopus species have a lower amount of mineral accessibility compared to the Ascomycota. And if we remember, the difference between the two phylum are the cell walls. And particularly for the Mucormycota, the cell walls are made of chitosan, which we know that they could absorb some of the minerals. However, this is still a hypothesis, and it will require a further study to investigate this hypothesis. We know that fungi have a very complex mineral homeostasis. They can grow at a wide range of mineral concentration, from a low mineral concentration to a very high mineral concentration. High amount of iron and zinc can be toxic for most of the organism. However, fungi have the mechanism to store excess amount of iron and zinc in their vacuole as polyphosphate. We hypothesize that this storage mineral as polyphosphate will have lower mineral accessibility. So we use the fungi Rhizopus oligosporus and grow it in a media with 0.2 ppm up to 21 ppm of iron. And as we can see, the iron content of fungi increase very significantly when they are cultivated in a media with high iron concentration, from 2 milligram at low concentration, and at high concentration, the iron content of the biomass could reach to 77 milligram of iron. But how about their accessibility? As our hypothesis, at low iron concentration, the fungi biomass have the iron accessibility of 100%. All of the iron are accessible. While at higher concentration, not all, even they have the higher amount of iron content, not all of the iron are accessible. For zinc, we can see that a similar trend. At low concentration of zinc, the zinc content is at about 3.7 milligram, and the zinc content of biomass increased very significantly as we increase the zinc concentration in the media. How about their accessibility? The completely opposite with iron. At higher zinc concentration, the zinc accessibility are higher, while at lower concentration of zinc, the zinc are not accessible at all. At low concentration, even they show some anti-nutritional properties for zinc. Then the question is, what happens if we cultivate the fungi that containing anti-nutrient? One of the anti-nutrient are phytate. Phytate can bind with the iron and zinc, make it not accessible. And in this study, we use oat, which contain phytate, as the cultivation media for the fungi. We hypothesize that the phytate present in the fungal biomass will most likely, maybe they will stay present in the fungi and affect the fungal mineral accessibility. But we can see that using oat flour, the phytate of oat flour is at about 1 gram per 100 gram, while the fungi Rhizopus oligosporus cultivated in the oat flour, it has a significantly reduced phytate content. And for the fungi in glucose media, they do not contain any phytate. However, will this improve the iron and zinc accessibility for the fungal? And we can see for the oat flour, it has 4 milligram of iron, and for the fungi, they have the higher amount of iron. And also for the zinc, it has higher amount of zinc compared to oat flour. How about the accessibility? For iron, we can see that the oat flour has a non-accessible iron because of the phytate. And for the fungi biomass cultivated in oat flour, it has a significantly improved iron accessibility. And however, for the zinc, we see that even after the fungi has lower phytate content, it still remains not accessible for the zinc. Another anti-nutrients are polyphenols. And we know that polyphenols could interact and precipitate proteins and minerals. Wine, one of the Europe's largest agriculture product, and in producing wine, it generates quite a lot of grape residue, and we would like to use the grape residue to grow the fungi. But then the hypothesis is, will the fungi accumulate the polyphenols from the grape mark? And if so, will it affect their protein digestibility and mineral accessibility? And the answer is yes, but it is highly dependent on the species. For example, for Neurospora and Aspergillus, after growing the grape mark, the total phenolic content of Neurospora and Aspergillus, they are similar, very similar compared to when they are grown in the glucose media. While for the Rhizopus delimar, a significantly higher amount of polyphenols are observed after when they are grown in the grape mark. And this affects their protein digestibility. If we see that the protein digestibility of Rhizopus delimar are significantly reduced, the protein are completely not accessible. This is mostly because of the higher amount of polyphenols. And so for the iron, for the Rhizopus delimar, we can see that even they show some anti-nutrition for the iron when they are grown in the grape mark for Rhizopus delimar. So in conclusion, we can say that fungi have a protein digestibility compared to that of animal-based protein, and fungi mineral accessibility are highly dependent on the media mineral concentration. And fungi cultivated in oat can reduce the phytate, but only improving the iron accessibility. And certain species of fungi can accumulate polyphenols from the winery side stream, and it could reduce their protein digestibility and the mineral accessibility. And so I will conclude my presentation. Thank you.
Thank you, Ricky, for your presentation there, and you showed us great insights from your work where you looked at protein and mineral bioaccessibility from filamentous fungi and how growing these in mineral-rich media or growing them on substrates rich in anti-nutrients can affect the bioaccessibility of these compounds. So now we are opening up to question and answer. So if you do have a question for Ricky, please make use of the question and answer box and we will get Ricky to answer your questions. In the meantime, I do have a few questions myself because I don't see any questions from the audience. So you showed us very clearly how the degree of protein hydrolysis for your for the filamentous fungi is more or less doing okay compared to the animal-based proteins. But I was just looking at the values there, and they seemed a little lower from what we would know of animal-based proteins. For example, you have between 60 and 80 as total degree of hydrolysis. Would you like to comment on that?
Sorry, one more time. Is it
just your absolute values there for the degree of hydrolysis for the other foods that you compared the filamentous fungus with? So you had your casein, you had your chicken, and your pork and beef.
Yes.
Yes. Would those values be a factor of the method you used, or would you like to give a comment on that? Yeah.
Because in the method I used for this INFOGEST, I reduced the amount of the amount of the pancreatin I did in the beginning of my study. I got then but I used the same method for all of my samples. So
nice.
yeah, that could be to compare. So that could be a possibility that this value will be different with another study, but at least with the same method, I can still compare with my fungi with the same sample. Yeah.
Oh, no, that makes sense as well. Thank you so much. I'm just going to open one question here in the question and answer. We have a question from Alejandra, and they ask, have you measured the content of beta glucan? Is it high and significant? It could be a good compound to have in the final product, they ask. So they want to know whether you have measured the beta glucan in your fungi.
I know, yes, fungi has the beta glucan for the Ascomyces, but I did not. Unfortunately, I did not do that for my work. I spend most of my time on the Zygomyces, which do not have the beta glucan. So yeah, unfortunately, I did not. But yes, beta glucan actually is a very interesting compound. And it could have a lot of effect even in the digestion and further for the fermentation. I agree totally beta glucan, but unfortunately, I did not measure it. Okay. Sorry. Shannon, are you muted or?
Right.
Sorry.
Just
Hello?
a second.
Oh, okay.
I think we are all sorted now. I was just saying, I hope I hope Alejandra is answered. Then we have another question from Mahmoud, and they ask, could you make it more clear about the impact on the digestibility in the presence of polyphenols? And maybe I'll let you answer that question because they've got two questions in one. So can you explain more about the impact on digestibility in the presence of polyphenols?
Yeah, like some species, they can accumulate polyphenols, the Ascomyces, like the Aspergillus and Neurospora. During my study with the grape mark, they do not, for the Ascomyces, they do not accumulate the polyphenols in the biomass. But for the Mucormyces, the Rhizopus, they have accumulate a significantly higher amount of polyphenols. And that higher amount of polyphenols cause the protein digestibility to be lower significantly, and also for the iron and zinc and the minerals. At the same time, it's a double-edged sword. Like polyphenols, we know is also have a benefit, but at the same time, if we want to use this fungi as the mycoprotein, it could have the detrimental effect. And I think, yeah, it's a trade-off that we need to take. But yeah, I only use the grape mark. I don't know what are not all polyphenols could impact hinder the protein digestibility. So that, but currently I only study with the grape mark. So
right, thank you. They have another question about the oral phase and why you increase the time to 1 hour.
Yeah, because I reduced the. I want to include salivary amylase in some of my study, which has the starch, like the oat, but salivary amylase, like, I think it's quite expensive, like human salivary amylase, like only several units. It costs quite a lot. So I think I want to reduce the amount so I can work with more sample. And by reducing the amount, like, then
like in the kinetics of enzyme. I was thinking that way, like by reducing the amount, then I will increasing the time, maybe to compensate that. But
Okay.
in the end, I realized it is not a good idea to do that, because my fungi also maybe could have this endogenous protease, like they already have protease by itself that could already react, like start hydrolyzing during that incubation. In the end, I don't think it's a good idea to do that, but that's what I was thinking before, like to reduce the amount of salivary amylase.
Oh yeah, thanks. Thanks for pointing that out,
Yeah.
because yeah, I'd noticed you had tenfold less the amount of enzyme. So
Yeah.
I think it's nice that you share how that maybe didn't turn out for the best in terms of other things happening at the same time. So thank you for that. We have Elizabeth as well with a question. Did you analyze the presence of chitin or chitosan in the fungi cell walls? And how would you access the bioavailability of the minerals? Are those not complexed with the food matrix?
Yes, I analyzed the chitin and chitosan, but I did not see. I just analyzed the cell wall content. Actually, it's in one of my study that I have not published about it. But I did not study how this interaction. Like I only measure the content, but how this interaction of this cell wall on the minerals, I did not. I think it really requires a separate experimental design. Yeah, I need to think how to design an experiment to answer that question. I have not. I did not try to do that, like answering that question. But yeah, I measure it's at about the cell wall content is about normally about 30% of the fungal biomass, and chitin normally like total glucosamine, like chitin and chitosan, the total glucosamine is about 50% of the cell wall material. So yeah, in that range amount.
Yeah. Yeah, thank you for that. We have another question from Chada, and they want to find out a little bit more information about how you determine your mineral accessibility. Just briefly.
Yeah, the supernatant, the digested, I centrifuge partially, then I just directly after centrifugation do the filtration with the syringe filter, and then analyze with the instrument. So it's only the solubility. I did not do any dialysis. I did not do any the cargo to cell. Yeah, only the solubility is a limitation, but I think it's for my purpose. Yeah.
Okay, thank you. I think it's also the same as Hanitra's question. How do you measure zinc or iron if you say not accessible? But I think you've already answered that by explaining how you did your work. Then someone is asking, which was the method to measure proteins? I suppose they're asking about the degree of protein hydrolysis.
The degree of hydrolysis is using the OPA method. The primary amine, and for the protein content, I analyze the amino acid, the amino acid profile. For fungi specifically, I don't recommend working with the crude protein. Because of the cell wall has a lot of nitrogen compound, and the component of the cell wall itself also can change by the cultivation. So there's no single conversion value that you can use for the protein content for fungi. So amino acid, if for the protein content which need to use the amino acid, then for the degree of hydrolysis, I measure with the OPA.
Yeah. Yeah, I think that was all the questions we have from the participants. Well, maybe just a final question from myself, Daniela. I don't know if you have a question as well. I will ask mine first, and then we can have a go. In the end, you show us how the polyphenols also influence the anti-nutrient activity or profile of the fungi. Like, how did you determine this? What was the specific anti-nutrient or activity test that you used?
Sorry, I don't get the how.
Yeah. So you showed with your polyphenol results how you just summarized it as anti-nutrient activity increased. Was there a specific anti-nutrient you were focused on? Which method did you use for that?
I did not measure the anti-nutrient. I mostly only see the amount of the compared to the blank, like compared to the digested, like the blank which do not have any fungi. So if I see that the compared to the blank, like the mineral content or mineral concentration in the blank is lower than the blank without any fungi.
Right.
So most likely then it bind it takes up the minerals that is supposed to be in the without any fungi. So that's why I indicate anti-nutrients by that way. To measure the anti-nutrients, no, I do not. It requires a different. I need to develop method. Yeah, I did not go through that area.
Right. No, thank you so much. There was one more question that just popped in now. And they ask, fungal matrices can contain high levels of phosphorus and sometimes phytates, the metabolites, and this depends on the substrate on which they are grown. So how did these components influence solubility and intestinal uptake of essential minerals like iron and zinc in your study?
It's a very good question. I actually also I did a separate study on that, like polyphosphate, like fungi has a high amount of phosphate. Is extremely high phosphorus. How this affect actually, I can say briefly or not much. The phosphorus. I have not published this study, but the phosphorus I add some polyphosphate, some alkaline phosphatase is actually do not have any effect, but it's not yet published. The result is not yet published, but there's an indicator. The polyphosphate, I did not show any. By adding alkaline phosphatase, at least it does not show any increase in the accessibility of the minerals, particularly minerals, because polyphosphate, my hypothesis, it could affect the minerals. So at least maybe it does not have any effect for that. But yeah, it will require more deeper study. But I agree, it's a very important question.
Perfect. Thank you, Ricky, for taking time to answer the questions from the participants. I think everyone is happy. I see a lot of thank yous there in the Q&A, so everyone is happy with the way you answered your question. I'll just hand over now to Daniela.
Okay, thanks. Thanks, Shannon. Thanks very much, Ricky, for this excellent presentation and for answering so many questions that we had today about your presentation. This webinar will be available on YouTube in the INFOGEST channel, as well as all the references to Ricky's published studies so far. We wish you all the best in the next steps of your career, Ricky. Thanks very much for participating. And we will be back next month on May 6th with a presentation by Mona Grunwald from Hanover University in Germany. So thanks very much, everyone, for joining today, and we'll see you again in May. Bye.
Thank you.